the spatial gradient in the myelin map, and we're going to key on one particular region of interest. This white outline represents the regions of a sharp gradient in myelin content, and we label it 55B as a marker for reasons that will become apparent in just a moment. But next we note that the task MRI contrast, the language versus or story versus baseline, lights up this little patch inside the 55B outline, and the spatial gradient shows it is completely surrounded by this gradient ridge. If we use resting state fMRI and put a seed location in one location, it shows strong functional connectivity with the region inside the box, and if we take a different seed, it lights up other regions that are related to eye movements, and the spatial gradient by this third modality agrees with the task fMRI and the myelin gradients, so all three modalities essentially give the same answer, and that's critically important to our approach. Matt and I call this area 55B because a half century ago, Adolf Hoff had identified a lightly myelinated region in what we consider to be pretty much the same point, and so we had to give it a tag. We chose history rather than completely novel naming scheme. And Matt was able to apply this general gradient-based multimodal approach to all the cerebral cortex in both hemispheres, and the result is this multimodal parcellation shown here, in which we identified 180 areas in the left hemisphere, a matching set of not perfect but fairly symmetric, same 880 areas on the right hemisphere. They're colorized here according to the functional association related to functional connectivity to identify visual areas in bluish hues, sensory motor areas in green, auditory in red, regions that are activated during tasks in white hues, regions that are turned off during tasks in resting state, the so-called task negative in dark, and we also gave these areas labels, some of them for regions that matched what's in the published literature from dozens of different studies. We were able to use the same published name for 83 different cortical areas, but 97 of the 180 areas we essentially gave new names, although in many cases it was splitting, for instance, TE1P versus TE1M and TE1A according to their finer grain parcellation of a published dark parcellation. So this is a group average parcellation. As I touched upon a moment ago, Matt was able to introduce what we call an areal classifier algorithm to identify each of these 180 areas in nearly all individuals, and I'll just show you an example of that in a moment, but I want to comment briefly on the data sharing aspects of this, which, as Jerry alluded to, has been near and dear to me. So we were able to generate a publication with many different figures using our Connectome Workbench software, and we take the data as generated by the software, generate fully annotated images, publish the images, and then we upload the data associated with that image to our database we call BALSA, and at each of the figure legends in the published online article, you just click on that URL, and that takes you immediately to our BALSA database, and in particular to the access point for the particular data set we call a scene file that lets you say, here's the image on a preview postage stamp size in the database. If you actually want it, you log in, click on a download option, and in short order you've downloaded to your laptop or computer at home the data set that lets you launch the visualization and see exactly what was in the paper and navigate it, rotate it around, or manipulate the data and make good use of it for whatever purposes you want. And you can get, for example, downloaded labeled parcellations if you want to try to better remember who's called what in this parcellation. The ability to take not only a group average that you see here, but individual subject parcellations I mentioned before, and that takes advantage of an areal classifier method whose exact operation I don't have time to spell out, but basically it works very well. Here's a parcellation in white boundaries overlaid on an individual subject's myelin map for one individual. Here's one particular area. The same area in a different individual has a somewhat different size, shape, and exact location, but it's identified robustly by the classifier, and it confirms our expectations that areas vary in size, shape, and precise location, but we have the data now to enable a variety of follow-up analyses. We can also take this data set and analyze resting state networks. So this is a study done by our colleagues at Yale and Princeton, the so-called Cole-Anticevich network partition. And so basically each of the colors here represents, for example in yellow, a group of areas that have similar functional connectivity and are strongly connected with one another and are organized into these resting state networks, of which there are 12 in this particular scheme. And this is a functional connectivity matrix that allowed Cole and Anticevich to identify these networks, and they are organized into networks related to sensory, vision, audition, so on, language, and other cognitive functions. And it's not shown here, but this analysis also gave the very useful parcellation of subcortical structures, hundreds of subcortical ones as well as the cortical maps. So this is a very rich addendum to the MAPS multimodal parcellation. So in the remaining moments, I want to touch upon two distinct themes, evolution and clinical issues, brain disorders. So each one will be relatively brief, but I hope you find interesting. Here is the human myelin map that you've already seen for reproduction of the human pattern. Here in the middle panel is the myelin map for the chimpanzee. Next over is the macaque myelin map, and then the tiny marmoset's myelin myelin map. So in each case, we can identify regions that are very likely to be homologous. The MT motion processing complex is one of the more striking, and so this gives us access to inferences about evolutionary homologies, and the lightly myelinated regions that are present in all species are at a glance much more prominent in humans compared to the non-human primates, and are particularly small over in the marmoset version. We can look at these evolutionary issues in the context of other approaches. So if we take our myelin maps for human and macaque and drill down on them and compare them by making a mapping between macaque and human that draws upon a set of colored regions here that are regions, cortical areas that are likely to be homologous across species. So this is a mapping between monkey and human cortex that allows us to estimate the degree of expansion associated with these different evolutionary lineages. So this is what we call a cortical expansion map in human versus macaque, and you can see the reddish regions are the regions of maximal evolutionary expansion in lateral, parietal, temporal, and prefrontal cortex that is dramatically greater than the very modest expansion in early sensory areas, and especially. This is a replication of this expansion map related to evolution that we're going to now put side by side with what we call a developmental expansion map. So remember, I told you briefly a little while ago that at term equivalent, the cortex is about 1/3 the surface area of the adult form, and if we look at a relationship between these two, we see that developmental expansion postnatally is not uniform, and the hotspots of developmental expansion are more modest quantitatively, but strikingly they agree in rough terms with the hotspots of evolutionary expansion. So regions that are associated with higher cognition are evolutionarily expanded. They expand preferentially in postnatal development. They are the lightly myelinated regions that you've already seen. And one last addendum that I think is quite nice, if we take advantage of the cellular level analyses of Guy Elston and his colleagues, they've shown that with selected sampling of early sensory areas, cell bodies and dendritic arbors are small in densely myelinated regions. In higher cognitive regions like TE and prefrontal cortex, the dendritic arbors are much larger and they're more synaptic density per cell. And so the idea is that regions of low myelin and have large dendrites, more synapses, and may well be associated with greater synaptic plasticity, although that's obviously speculative. Jump shifting over to our last major topic, brain disorders. Just in broad strokes, everyone either has a disorder or knows somebody with one. That's just the unfortunate reality. It has enormous social and economic impact. Brains malfunction in an incredibly diverse number of ways. We're far from having cures, and treatments that we have are of limited efficacy in most cases. We don't understand what's gone wrong, and what we need to advance the frontiers are a combination of better basic neuroscience in humans and animal models, greater acceleration of technical advances such as those that have already made progress with the BRAIN Initiative, better translational neuroscience, improved diagnosis and treatment, and I'm going to give just two quick examples in the remaining moments. So one is clinical depression, which is all too common. One way of treating severe depression is with deep brain stimulation of the subgenual prefrontal cortex, roughly here. Another way that's emerged that has some potential but is still early days is transcranial magnetic stimulation, putting a magnetic stimulating pulse above the head in various locations. And it turns out that works well in some individuals and not so well in others. And a recent study by Drysdale et al. Has shown that diversity in functional or clinical responsiveness is related to brain circuitry as inferred from resting state functional connectivity analyses. And the hope would be that better, more careful analyses of resting state functional networks in individual subjects may improve the specificity with which we both stimulate appropriate regions and interpret the data. So that's a way forward for the future. Jump over to autism, which is obviously of high interest to the Simon Foundation. Some studies report what they call hyperconnectivity using functional MRI, greater inferred connectivity. Others report weakened connectivity, hypoconnectivity of particular networks. A very recent study from Udin's group reported no differences between functional connectivity across comparing with autism, typical development, and ADHD. And they interpreted this as likely more plausible interpretation relative to the false negatives and false positives reported in earlier studies. But what I want to draw attention to is that the methods used in most of these studies are what I would call the traditional paradigm that I've already critiqued. They are based on rather short scanning data, non-optimal analysis methods, and the HCP style paradigm that I've already introduced, I claim, and with considerable evidence to support it, is fundamentally more sensitive and a better approach. So I'm not going to walk through these again, but just remind you there are better ways of going about business. And the hope is that doing this in healthy individuals, such as the Lifespan projects in aging and development that are currently underway, the adolescent brain and cognitive development is a parallel study that's getting over 10,000 children that will be followed for a decade. There's a baby connectome project and a developing human connectome project that are using cutting-edge methods. There are disease connectome projects that are currently underway that will look at a number of disorders. Unfortunately, autism is not yet one of them, but hopefully something pushing the envelope on autism will also emerge. And to wrap it up, what are the most important bottom lines? Cortical parcellation is fundamental for understanding brain organization. We have evidence for 180 areas per hemisphere larger in human, smaller numbers in animal models. Cortical connectivity shows a very complex distributed hierarchical organization with a tremendous range of connection strengths. We're far from having a human connectome that spans the macro through the meso and micro level. Evolution has given us evidence for preferential expansion, particularly associated with higher cognitive regions, and HCP-style studies of disease development and aging are critical for understanding and improving diagnosis and treatment. So maps are key to understanding for both Earth and brains. Better maps make for better analyses, all the way up to satellite imagery, structural and functional MRI. Google Earth has been transformative for our understanding of navigating the Earth, and connectomics and the associated methodology improvements will hopefully accelerate progress in studies of brains. Everything I said relied tremendously from contributions of literally hundreds of individuals, HCP Project and more. I highlight a few names, Matt Glasser in particular, Deanna Barge, Camille, Steve, and Dan, and people in my lab have also contributed greatly. Funding has been crucial, and the clock has run out, and I hope there's time for a little bit of discussion and questions.
Structures and also for dynamic systems, there's a way to find order even if you don't know what the order is. And sort of to illustrate the basic idea here, we can look at Pollack and Mondrian, and it's clear to everyone that which one is more ordered. Well, it's clear that the Mondrian is more ordered, I hope. Okay. The question is, can we quantify it? How would you quantify it? Okay. And as in everything nowadays, you expect the answers probably on your cell phone or your computer. Okay. But it's not on Google. It's in your camera. Okay. Because the camera stores these images in a compressed form, and if you're lucky and you pay enough money for your phone or your camera, it losslessly compresses it, which means you can get all of the information from before the compression back by decompressing it. And when you do this, you find, aside from overhead, so these are 3 images all the same size. One's the Mondrian, one's the Pollack, one's just white. There's overhead in the compression, which corresponds to about 9,000 bytes here. And if you just look what it does with the Mondrian, it's only 12,000. Subtracting the overhead, it's 3,000. Subtracting the overhead from the Pollack, it's a megabyte. So you can tell from that directly, quantitatively, that the Mondrian is more ordered than the Pollack. Okay? Quantitatively, a factor of almost 300. Now, the basic idea here that I want to get across is that the more organized, ordered something is, the less information you need to describe it. That's the essence of the idea. And the question now is how you find out how much information there is. And there are several ways of doing it. The original way, the start of information theory, was Shannon's work on Shannon entropy. Shannon entropy is the same as thermodynamic Gibbs entropy. Okay? It's just the probability for an occurrence times the log of the probability. And that's a probabilistic combinatorial way. That's the way we learned it in statistical mechanics. But Kolmogorov, same Kolmogorov that gave us the turbulent cascade, same Kolmogorov that made sure that the Russians are much better in math than we are because he wrote the textbooks for high schools and public schools in mathematics. He came up, he's a very original guy, came up with another way. He said, okay, you can do it probabilistically or combinatorially, but you can also do it algorithmically. And so what he said is you can measure the information in the system by the minimum length of a computer code which will generate that data set. Okay? We'll look more at this later, but that's a great idea. It really allows you to think about problems a lot. Clearly. The next thing you can do is you can look at lossless compression. So lossless data compression is a way of trying to approximate what the Kolmogorov complexity is. It's trying to reduce the information to the smallest data file you can from which you get the complete information back. So if compression algorithms are great, then what you get from the length of a compressed file from the data set, okay, is the entropy essentially. And why might this work? The reason it might work, if these things are great, is because everything you do nowadays, communication, computing, stock trade, cell phones, streaming video, data storage, everything depends on data compression. And the people in information theory and computer science know that, and they know what the limit is. The limit is the Shannon entropy, so they do the best they can. And this is worth something like, information is worth like 4.8% of the GDP of the world, and there are other things, there's finance and other things that depend on it. Okay? And that's why you might hope, okay, and that's what we're experimentally going to test, okay, that the compression algorithms are really good, and hence give you a measure of the information in the entropy. Okay, the people who are really involved, really responsible for this work, all the work is really done by Stefano, who's a postdoc who works with Dov Levine, who's at Technion, and me at NYU. Stefano's at NYU. He actually is out in Illinois today. He got a job offer from University of Illinois, and he's really worried because he was raised in Italy, and in London, and in Cambridge, and he's worried he's going to be stuck in Urbana the rest of his life. Anyway, and Dov over here is the guy you guys may or may not know who invented quasicrystals with Paul Steinhardt. Okay, so from what I've told you now, you sort of maybe get the idea that I'm going to do something rough, not quantitative, I'm going to wave my hands a lot and stuff like that. And it's not true. What I want to show you is you can really do quantitative things with the data compression. And so the easiest thing to do is just show you an example. This is a dynamic model. It's a conserved lattice gas. What you do, it's in one dimension here. You can do it in n dimensions, but here's a one-dimensional version. You have sites which are occupied or unoccupied, only unoccupied or occupied, never doubly occupied. You can throw particles down randomly on this, okay? And the rule is if you have a nearest neighbor, then these sites with nearest neighbors are active. And what you do to the active sites is in the next step, you empty them to neighboring. You empty those sites to neighboring empty sites. And this gives you a dynamics, okay? And the system evolves with time until either it comes to a steady state where everything is still moving around because it can't find a configuration where you have no nearest neighbors, or it finds an absorbing state, okay? And then it stops evolving. So when you throw the particles down, okay, you can write down, in this case it's very easy, just zeros and ones, a data string here, and you can compress it. And as a function of the coverage of the density here, from 0 to 1, okay, you can look at what we call the computable information density, CID. It's simply the size of the compressed data file over the size of the raw data file, okay? And when you do that, you get this curve, which actually is just the entropy of mixing for a system like that. Turns out it does that pretty well. What was more interesting is when you let the system evolve with time, it does this, okay? And it gives you something which has a cusp here at a half, okay? Now, if this were the entropy, that would indicate that you have a second-order phase transition here, okay? So that's one thing you can do with this. But you can answer other questions as well. And the questions you might ask about active systems, about dynamic systems, not in thermodynamic equilibrium, is whether you can tell something about the ergoticity of the system, how many states are sampled, whether it goes organic, whether they're sampled evenly and things like that. We can do that with this. What we can do, for example, is if I just take the curve I just showed you here, okay, the final curve, this is after 10 to the 7th steps, okay, these were absorbing states, okay, on this side, less than 1/2. Greater than 1/2, these are all active states. That's obvious because at greater than 1/2, for the simple problem in one dimension, if I have greater than 1/2, I can never find something where I don't have a nearest neighbor occupied, okay? So these are the absorbing states. I can also ask, given the rule here that absorbing states are states where I have no nearest neighbors, I can ask, what are how many states do I have where there are no nearest neighbors? I can actually calculate that, or I can simulate it. I can just Monte Carlo, throw down particles and find all configurations with no neighbors, and compare that with what you get for the absorbing states from these dynamics. And you see that it's lower here. The absorbing states that are found by these dynamics are lower than all the absorbing states that there are. And from the idea that I'm trying to get across to you, that implies that these are more ordered. And we don't know what the order is, or we didn't know what the order is when we started. Once you know that these states are more ordered than these states, you can look at the states and see what goes on. In fact, what you can simply do is the two-point correlation function, the density-density correlation function, and what you find is you pick a particular density over here, you compare the correlation length, which is given by the decay of this correlation function, for all of the states and for the absorbing states, and you find there's a characteristic length scale over here, where they're correlated, and in the absorbing states, here it's less than 100, here it's about 100 times bigger. So here, once you know to look for order, you can go and look for it, you can find it, and you can find that it's simply in a correlation length for this alternate lattice, alternating sites. The reason it's more correlated, it turns out, is the dynamics here spreads things out. When these guys, it starts, let's say, in this dense region and spreads out from there, okay? And that's why it's more correlated. So you get something from this. You find order where you didn't know it was before. What else can you do with it? Well, another thing you can do with it, you can look at the time evolution of the CID, of the information here. And what you find is, if you start at low density, okay, it takes a short amount of time to find an absorbing state, okay? The higher the density is, the longer it takes to find that. You can plot the time that it takes to find an absorbing state as a function of density, and you find there's critical slowing down, just like in a second-order phase transition. And even just from looking, from fitting the CID as if it were an order parameter for the system, you can even pull out the critical exponents, okay? You can also ask about ergodicity in the system on the active side. This is the active side. Here we're showing on the active side all of the active states you can have that are allowed by the dynamics and what you find, okay? And they're right on top of one another, so all of the active states here are completely ergodic. And then you look, after doing all of this, just by compression, you look at what's been done before, and what's been done before is, for this active system, the order parameter is the activity itself, and in the absorbing states here, the activity is 0, and here the activity turns on, okay? And it's exactly at the point you find from doing the compression, and just the compression. Okay, so hopefully I've shown you that we're not just fooling around. We can actually do things with this, okay? I should mention that there's another group, Roy Beck's group in Tel Aviv, which came up with the same idea at about the same time, but they're using it to get the entropy for protein folding. Okay, information, order, and entropy. Okay, so Shannon started the field. Here's the Shannon entropy. It's combinatorial or from probabilities. The problem with this, you might say, why don't we just do this? So what you have to do the Shannon entropy is you decide on a certain block size, okay, and you go and see the different configurations that are in that block size, okay, okay, in your system. If you choose a block that's only 20 long, most systems you have won't sample. You don't have enough information to sample all the states that you find in that block. So it's very hard to calculate that from any experiment or even from simulations. Okay, you can do it if you know what's going on. For instance, if you know what the statistical mechanics is of your system. But we want to look at dynamical systems where we don't know the statistical mechanics. They're not in thermodynamic equilibrium. So this is meaningful for stationary processes, but it essentially needs an infinite ensemble. Here's the Kolmogorov complexity. Again, the length of the shortest computer program that yields the sequence that you're after. This has a couple of problems. Okay, first, shortest. How do you ever know that you have the shortest computer program that will generate that data set? Secondly, there isn't any general way, if you take a string, which is like taking, let's say, a rational number, an irrational number, I should say, take an irrational number and find a way to code that in a finite length code. Okay, if you could do that, if you find a finite length code to describe an infinite sequence, okay, then the entropy in that sequence is zero. Right? The information you describe it is zero compared to the length of the sequence. So this has two problems. You don't have a general way of finding the algorithm, and even so, you can't show that it's shortest. Okay, so what you do instead is compression, is use data compression. And one of the coding methods that's used most often now is Lempel-Zeev coding, which has many important characteristic. It's asymptotically optimal. That means asymptotically, it's been shown mathematically that it approaches the Shannon entropy, and the time it takes to do it is order n, order the length of your system. It essentially takes one pass on the system. I'll go quickly through this. So there are lots of different compression algorithms. The first one I ever learned as a Cub Scout was this one, which is Morse code. The way Morse code works is it looks at the alphabet, it looks at what is used. Actually, is there water around or something? Okay. It essentially looks at the usage of letters, and the most commonly used letters are E and T, and it gives them the shortest code, right? A dot for an E and